Hello, in this video we will talk about testing homogeneity of regression slopes in ANCOVA using R. So in the previous videos we have shown how to implement ANCOVA and ANOVA in R. So we know ANCOVA is a type of ANOVA that includes covariates in a function, because in addition to group difference for the treatments we are interested in, there are also covariates, and usually they are continuous variables that have effect for the dependent variables. So we have to include them in ANOVA function. And there's assumption for ANCOVA that assume the homogeneity of regression slopes. So what this means? So for example, in a data set called litter is a data frame. So we have several variables called weight, gestation, and treatment. So weight is a continuous variable. It is a dependent variable we will measure, and treatment is a category of factor variables for different treatments. And in ANCOVA and ANOVA, we want to test if the mean difference for the weight are the same for each treatment groups. So this is a very easy understanding, right? And gestation is a continuous variables act as a covariate, so included in ANCOVA, because this variable has also effect on the weight. So we have to include this one in ANOVA function, correct, right? So this kind of analysis is based on assumption that there is homogeneity of regression slopes. So which means for each group, for the treatment groups, the gestation on weight has almost the same regression slopes. So their effect on the weight are the same for each treatment groups. So if it is not, because there are other factors we have missed, right? So this is a very important assumption in ANCOVA. So we have to test this assumption at the same time we carry out an ANCOVA. So how to do this? In R programming, if we want to do this, we have to include the gestation and dose. Dose is the same as treatment group, this variable. So we have to include the gestation and dose interaction term in ANCOVA model. If the parameter for this part is significant, so which means the assumption of homogeneity of regression slopes is rejected, because their interaction has effect, right? So next we will show how to implement this in RStudio. First, the same as before, we load the library multi-comp, because the data frame litter is included from this package. So we show several observations from this data frame called the litter, with the dose, weight, gest time, and we do not use this variable currently. The 3 columns, and we also use a dplyr package to show more clearly for the information by group. So by those group, there are different values, their treatments from those, and their observations, their mean values, and standard deviation. So you can see mean values for this group without those are very large, and compared with other 3 groups with those. So next we can do an ANCOVA using AOV, weight, then tilde symbol on the right side. We just add gest time and dose and with data. So the result from ANCOVA shows both gest time and dose are significant. So which means if we control gest time then those have different effects on weight in different groups, right? So now we will include part of the interaction in ANCOVA to test if just time the effect on the weight is the same for different treatment group from those variables. So we can do an ANCOVA again, but this time on the right side we set the expression as just time multiply or asterisk with those. So this kind of expression, if you want to check the AOV function in detail, this exactly include 3 parts. The first part and the second part are the same as the last time we do ANOVA in this function. These two. So, but this time, in addition to these 2 variables alone, there is an interaction part also included. So this multiplication include odd first power, second power, first power for these 2 variables and interaction of these 2 variables. So we will run, then we show the results of this ANCOVA. So you can see in this ANCOVA results, there are 3 variables. Just time, those both are significant, same as before, and just time, then colon those, this is the interaction part. And this part is not significant because the p-value is larger than 0.05. So which means we can hold the assumption that the effect from just time on weight are the same for different treatment groups from those variables. Thanks for watching.